So, sorry, I need, the system is started already. Okay, so welcome, ladies and gentlemen, to our webinar, which is organized by the FooD-STA project and in cooperation with the ISEKI-Food Association, and which is also supported by the by Elsevier. Today our topic is Bacillus cereus from a food industrial perspectives, and the speaker is Florence Postollec from ADRIA in France. And my name is Gerhard Schleining. I'm BOKU from the University of Natural Resources in Austria, in Vienna, and I'm also coordinating this project. The program of today is I will give you a short introduction how you can participate to this webinar, how you can also interact. Then I will have 2 or 3 slides about the project to give you an idea about the project. Then we will have the presentation, and then you have the possibility to ask questions. There are 2 options. You can write your questions into the chat box in your control panel. I will show you later, or you can raise your hand to do that. After the webinar, you will also receive an email, probably by tomorrow, where we want to have feedback by you about the quality of this webinar, and also you can then suggest further topics for future webinars. You can also send us anytime your suggestions for future topics to this email address. So the control panel should look similar to this. It depends on which operating system are you using, but in general you have all these features available on this control panel. You have a box where you can post your questions. This is the on the lower part. Then you have a red icon with an arrow where you can minimize your panel if it's in front of the presentation. Then you have the feature to switch to full screen, and this is the important icon. If you click on that, I will get the signal that you have raised your hand and that you want to tell us something, because all the microphones are muted during the presentation, and when you raise your hand, I can open your microphone. I see that there is one person, Teresa Garcia. She is connected, not connected with the microphone, but she has maybe the wrong settings. So you should, in the settings, you should switch your settings from telephone to microphone and speakers to enable that your microphone is working. So if you want to raise your hands, maybe you can try to click on the icon that I can see it is working. Okay, I see some people raising your hand. If you click again, then you put your hand down. Okay, this is working. Thank you very much. I also have to tell you that this session will be recorded. That means people who have who missed this webinar, they can also have a look afterwards, but they cannot interact with the speaker. But they can also listen what was discussed in this webinar. Okay, this activity is a task of the European FooD-STA project. This is an Erasmus Plus Knowledge Alliance project running from 2015 to end of this year. It's coordinated by BOKU, and we have several partners from universities and several partners from industry. It's a It's a knowledge alliance project where we want to establish a training alliance between food industry and also universities teaching in the food sector. The vision, what we have when we submitted this proposal was that, as most of you may know, that we have on the one side usually the universities, and on the other side there is the food industry, and usually we have different goals. The universities, they're focused on research, they are looking for fundamental mechanisms which are behind things, they focus on publication, whereas the food industry wants to keep the knowledge. They are very focused on intellectual property rights. They also are not very much interested in what is going on. They want quick solutions in many cases, and they have also time constraints for research and training. And with this project, we want to close this gap by establishing a so-called FooD-STA Center, where we want to bring together these both worlds. And we have already learned that it is important to establish long-term partnerships on European level, and also to start with simple goals and not to have too high expectations. The advantage of this project is that we want to extend the collaborations between universities and food industry on a European level. In a local level, there is already some collaborations, but we want to extend this on a European level. That means that members from Spain can also have access to the research facilities in Germany, for example, which they don't had before. So the consortium of this project, we have 7 universities, we have 3 food companies and 11 multipliers and training providers in these countries, which are marked yellow on the map. From the universities, we have BOKU, then in France we have Agropolytech. In Porto we have 2 universities, University of Porto and the Polytechnicum in Quimper. Germany, University of Hohenheim, in UK, University of Leeds, and in Italy, University of Teramo. From food companies, we have 2 medium and big-sized companies. One is located in Portugal, but they have also sites in 4 countries where they are producing fruit preparations, mainly designed for dairy and bakery industries. And GP-Food, which is located in Spain, but has also other facilities in other countries and also outside of Europe. And they are producing mainly ready meals products. We have also Nestlé on board, which is not a full partner, but associated partner, because it's located in Switzerland, so it's not funded by the European Union. But we have a person who is our contact there, and we get information from Nestlé, we get input, and also they help us for disseminating results. What is also very important for this project is the contact to the small and medium companies, because the food industry mainly consists of small and medium companies, and therefore we have the food associations on board from different countries. In Austria we have LBA, in France we have ADRIA, in Italy Federalimentare, then in France ANIA, and in Spain FIAP, in Greece CEFT, in Portugal FIPA, and associated partners, which are associations linked to the teachers. For example, the ISEKI-Food Association, which is also supporting this activity by providing the tool for having these webinars. And it's also a network for university and teachers. And we have also associated partner EFOST, the European Food Federation Network, which is more industry related. And we have also some associations which are related to the university administration for exchange of students and teachers, the Eroica network, and also space. Our main outcome is this European FooD-STA Center, which is an independent platform with physical hubs in different regions. So we have one in Vienna, we have one in Hohenheim in Germany, Stuttgart, we have one in Essence, in Spain, in Barcelona, in Italy, in Teramo, in Porto, in Leeds, and also in Paris. And these local hubs will be the interface to the local industry and to the local universities to involve industry also in the food study programs, but also to train people working in the industry for continuing education. So this was a brief introduction of the project. You see here also the website where you can also find more information, and this network is also to be extended. At the moment it's for free, so you can also apply to be a member of this network and to have access to our materials. Just go to the website and you will find information what benefits you have and how you can subscribe or register for this project. It's very easy just to fill in the web application form and then you will get an approval. Coming back now to our webinar. At the moment I see we have 16 people attending this webinar. Also 61 people have registered. Registrations are coming from 25 countries, also from outside from Europe. There are people from US, Brazil, Ethiopia, Kazakhstan, Thailand, Vietnam, down to New Zealand. So I don't know if all the peoples where the people are, but I can check later on where the participants are coming from. But anyway, our webinar today is given by Florence Postollec on the topic of Bacillus cereus, which has the ability to form endospores, and she will has 3 sessions in this webinar. Case studies following to give more insight in the industrial applications for the biodiversity, about the methods which are used to address the diversity and to distinguish phylogenetic groups. Then about germination and growth ability, and also about the inactivation by processes. Florence Postollec is project manager in ADRIA. She is biochemist and obtained a PhD degree on bacterial interactions at the Faculty of Medical Sciences in Groningen. She gained also experience on molecular microbiology when she joined ADRIA in 2005 as a postdoc, and she, within the frame of a competitive technological cluster, she is also collaborating with the MAFAD team on risk associated to food-borne spore-former contaminants along a 10-year research and development axis. As a project manager on food quality and safety, she is particularly active in setting up projects, coordinating multiple stakeholders, and dissemination of results, especially about mathematical models to predict the bacterial behavior to optimize process and adjust food shelf life. So this was a brief introduction. I will give now the floor to Florence. Just something to mention. This series of webinars is also supported by Elsevier. They will offer to promote the presenter by giving access to 2 publications of the presenters for a free download. So here is the link. You can download these publications for free. I think for 2 weeks from now you can have these options. So this is the contribution of Elsevier to support also our presenters. Now I will give the floor to Florence. to start her presentation. I will make her presenter. If you have any questions, you can, in the meantime, maybe raise your hand so I can answer your questions if everything is working, or maybe there's a problem with the audio. But as there is a question from Marcel twittering, so maybe he put the hand down. But there is a question from Rebas Koi. Maybe I open shortly the microphone so this person can talk. Hello, Rebas Koi. You want to ask something?
No, sorry, no.
Okay, it was a mistake. Okay. So then I will give now the floor to Florence, so maybe you can open your presentation and share your screen. At the moment, we don't see your screen. Okay, yes, it's working. Please, the computer is yours.
Okay. Thank you first for this nice opportunity to present some work that we've been performing on spores, more particularly Bacillus cereus, and I entitle my speech Bacillus cereus from a food industrial perspective, because this is a wide range topic, and I would like just to focus on some state-of-the-art information that could be important to share with food industrial in order to assess risk associated to Bacillus cereus. My name is Florence Postollec. I'm from ADRIA Food Technology Institute. ADRIA is part of ACTIA, National Coordination Structure. So via this membership, I'm glad to participate to this webinar series. We are about 50 persons working on food quality and safety issues. We have close link with food industrial. Part of the employees are doing audit, training in food industries, and the other part is working in the lab, molecular and microbiological lab, and food safety, food science, characterization lab as well. We are performing different advices and services to food industries, for instance, validation of alternative methods. In food microbiology, we also develop some molecular tools, targeting and typing foodborne contaminants. We also perform challenge tests in food to validate formulation, process or shelf life. And we have, as was mentioned by Gerhard in the introduction, a research axis with our university partner since now about 10 years on risk associated to spore-forming bacteria. So both regarding food safety and food quality. So we are located in the extreme and far west of Europe, as shown on the map. First of all, I would like to share with you some points in order to give you a short introduction of the topic. Bacillus cereus is Bacillus species, are spore-forming bacteria. I'm trying to change a bit my view. Spore-forming bacteria, which are ubiquitous and exhibit a wide range of diversity in behavior. Spores show high prevalence in raw materials, ingredient, and spores represent a dormant but highly resistant contamination of Bacillus species. In food, when conditions are favorable for growth, germination, outgrowth, and cell multiplication will occur. And for specific strain, that might lead to food spoilage and food poisoning issues. So as you see up, these points explain the persistence of spore formers in industrial plants and also yield to huge economical losses. Bacillus cereus may be involved in food spoilage. Food spoilage is regarding food quality. This is the deterioration of food texture or sensory attributes that renders foodstuff no longer suitable for human consumption. And Bacillus cereus may as well be involved in food safety, and this is the major issue associated with Bacillus cereus. Some strains have the ability to produce emetic or diarrheic toxin that are yielding to food poisoning outbreaks. There are a few fatal cases, but there is an increased trend of food poisoning issue, and this is representing in Europe more or less 5% of the outbreaks that have been reported. Food poisoning mainly occurs with takeaway or catering restaurant, and this is mainly due to temperature abuse or improper handling or storage of cooked food. But facing the trend of and lifestyle of healthy, attractive and convenient food, the market of processed, ready-to-eat food is boosting, and we might foresee increasing issues regarding food quality and safety associated with Bacillus cereus. So this is really a high topic at the moment. Regarding an industrial perspective, Bacillus cereus strains are ubiquitous. They resist to physical and chemical treatments. They may grow at refrigeration temperature. There is no food safety criteria in the European Regulation 2073, but only a process hygiene criteria, and this is only for dried infant formula or dietary food. And this level is ranging between 50 to 500 CFU per gram. But usually it is well recognized that a level of 3 log contamination is used as an alarm, a threshold to screen raw material and end products. So how to assess risks regarding Bacillus cereus hazard? So during my presentation, I will focus on 3 points because this is, as I said, really broad. So I just would like to focus on these points. The first one is regarding the lifestyle, the life cycle, which is highly complex due to the ability of these bacteria to form spore and recover vegetative growth. The second point will be regarding biodiversity. And the third one will be regarding methods that are available to screen or control contaminations. First of all, the life cycle. Spore formers have the ability to form spores. Spores are these highly resistant states of the bacteria. Spore contamination occurs in raw material, ingredient, environment, surface. This is yielding to issues regarding process and sanitation. Most of the time, conditions in which the spores are formed are not known. On the contrary, the germination process will occur in food. So the idea in food formulation and process is to avoid spore germination, outgrowth, and cell multiplication in food during shelf life. And this is regarding food safety and quality issue, of course. Really quickly, different process in this life cycle. Sporulation, which is the formation of one spore out of one cell. So you see here on this observation, microscopic observation, this is just a phase contrast observation. There is no need for staining because spores appear as refringent structure here in one cell. So you see here the spore, the endospore, and here the cell. So one cell will give one spore. Spores are dormant but highly resistant state of Bacillus. They are mainly, sporulation mainly occur when unfavorable nutrient conditions occur, most of the time when you have nutrient depletion. This is a micrograph of Bacillus anthracis and the spore. Since this bioterrorism attack, there is a lot of micrograph available on the internet. So you see really complex structure where you see here the core of the spore, where you have DNA and all the material, the compacted material of the bacteria, and then you have different structure, membrane, cortex, coat protein, And the last one is the exosporium. This is kind of balloon which is occurring when you are producing spores in the lab, but obviously in industry where you have a different kind of condition, mechanical shears and so on, this exosporium is not remaining. So there is a lot of research going on coat protein and cortex in order to identify biomarkers that could explain this resistance. Sporulation is a well-described cycle, so I'm not going into detail to this on this cycle because a lot of reviews already exist on this. This is clearly described how the pre-spore will acquire progressive resistance to UV, chemical, radiation and heat treatment before becoming a mature spore and being released in the environment. Spore may remain dormant for extended period of time, even though there is no metabolic activity, spores are still able to monitor the nutritional status of their surrounding, and they can rapidly respond to the presence of appropriate nutrient by germinating and resuming vegetative cells. And this is a kind of strategy that Bacillus species got to escape unfavorable conditions via dormancy. So when environmental conditions are favorable for growth, you will have outgrowth and multiplication of cell. So here is the short video that I want to show. These are not ours. This has been performed by Sandra Stringer from the IFR Institute in the UK, and this is a really nice video showing how the spore is losing refringence, the cell is popping out, and then you have vegetative multiplication of cells occurring. So of course this is a video, this has been performed in really controlled condition under the microscope, but this is really a good illustration of this phenomenon, this rapid phenomenon of germination. So once germination occurs, the spore lose the resistance of this structure, and then you have exactly the same condition as normal classical vegetative cells. Just to draw your attention on super dormant spores, which requires a combination of factors to activate germination. Most of the time only one germinant, one nutrient is enough to initiate germination, but in these really specific super dormant spores you need several germinants or combination of germinant, the presence of germinant and heat treatment to activate the germination of these spores. This is quite interesting to mention because in food industrial condition where you might have a successive stress, you can also have some issue related to super dormant spores. And there is a lot of publication and research going on germinosome, temporal gene expression to know a bit more about this process. So apprehending this, the life cycle of this bacillus is really important because there is a strong impact on the environmental conditions that the spore and the cell will encounter, and this will lead to some issue regarding resistance and germination. Depending on the condition encountered before, during sporulation or germination, you might have different kind of scenarios. So it's quite important to have an idea on this issue. I'm going to quickly show you some results that we acquired in the lab. This has been reported as well by other research team, but just to illustrate the impact of sporulation condition on spore resistance and spore load production. The thesis of Eugénie Baril was dealing with the sporulation and how to apprehend sporulation condition. So she worked on Bacillus weyenstephanensis, KBAB4 strain, which is a psychotolerant strain, and she produced spore at different temperature. And as you see here on this graph, where you have here the population of spore as a function of heat treatment, you have spores produced at low temperature, 10 degrees, that are being destroyed more quickly and easily by heat treatment of 90 degrees as compared to spores produced at a higher temperature, 30 degrees in this case. So the impact of sporulation condition on spore resistance is well documented, but this is quite important to address here because this is also comforting classical lab practices. Indeed, when you want to perform a challenge test and inoculate bacterial spore, you need to produce these spores, and in the lab what we do most of the time is just to inoculate agar, wait a bit, and then due to nutrient depletion the cell will become spores and you will have the production of spores. And these conditions are most of the time performed in optimal growth condition because you want to have a quick production of spore. And this is these results comfort and increase robustness of these practices because the spore that will be produced in the lab will also be the spore which will maximize the risk because they will be the most resistance obtained. Another PhD study performed by Clément Trunet was investigating the impact of process on germination abilities, and he used in this case flow cytometry, where you have here a representation. So you will just see the spores population that have been obtained, produced in optimal condition, and you will see the spore that will are growing cells and then cells when placed, when used in recovery, optimal recovery media, which is 30 degrees and neutral pH. For exactly the same condition, but when your spore will first be exposed to a heat treatment and then put in recovery medium of 30 degrees, exactly the same recovery medium, you will see that the process, this heat treatment, will strongly impact the germination ability of the spores. So we'll just click here and you see the spore population, which are becoming outgrowing cells and then cells. So all the spore population are now cells. In the other case, where you have a heat treatment before putting the spore in a recovery medium, you see that the spore population is more broad. And here you have the outgrowing cell and part of the spore that was inoculated in a medium reach the final cells development. And here you have intermediate states that are remaining or not in this state. This we clearly cannot see, but what is interesting to see here is that as soon as you have a process, you will have an impact on germination ability. And most of the time you have a wider range of heterogeneity of distribution. And the video, the nice video I showed you performed with Sandra Stringer, also reported the same result, a strong impact of process on the distribution and the heterogeneity of the responses after heat treatment. Last but not least, Émilie Gauvry is performing her PhD in the lab at the moment, and in March this year she presented some mathematical models which are taking into account the whole life cycle of Bacillus, using growth cardinal value, which are minimum, optimum, and maximum values for temperature, pH in particular. You are able to account for the whole life cycle of Bacillus. The use of cardinal values is well known for the prediction of vegetative growth, but using these values, you can also account for sporulation and germination steps, which are really specific to Bacillus. And this concept is quite generic because it has been validated in several strains of Bacillus, psychotolerant, mesophilic, but also thermophilic strain. So really nice result that I wanted to share with you today. The second part of my talk is regarding to diversity, biodiversity. If you look at datasheets that are available in Europe or elsewhere, you have the following information regarding growth limit, where you have minimum and maximum temperature that is allowing growth. Minimum and maximum pH that is allowing growth of Bacillus cereus, as well as water activity. So most of the time you have safe food regarding Bacillus cereus hazard as soon as you have a low pH and a low water activity, because in this condition the growth of Bacillus cereus is not supported. You also have information on toxin production, heat resistance, and guidelines regarding the handling and storage condition of cooked food. So for regarding Bacillus cereus food poisoning issues, this is really important to have a rapid cooling of cooked food and to have proper temperature storage, which is below 4 degrees or higher than 63 degrees, in order to avoid growth of this contaminant in the food. Bacillus cereus group, we mention, it is usually mentioned as a group, presumptive Bacillus cereus group, or Bacillus cereus sensus lato. This is a group of strain which are belonging to these different species, which has been reported in the literature. So here I indicated the date and the author that published information regarding these different species. So you see, Anthracis is quite known since a long time, and the last two are quite are brand new members of this B. cereus group. Bacillus anthracis is involved in anthrax. Bacillus sensu stricto and Bacillus cytotoxicus are both involved in food poisoning. Bacillus thuringiensis is used as microbial pest control. Bacillus mycoides and Weyenstephanensis have the ability to grow at low temperatures, so they might be involved in food spoilage of refrigerated, ready-to-eat food. And Bacillus thuringiensis, which is the last reported microorganism of this member, is a probiotic. So you see a wide biodiversity of Bacillus cereus, and they might be involved in both food quality and food safety issue. So if we draw the parallel with these Dalton, you know, these Dalton fictional outlaws, they have been created by Maurice and Goscinny, and they appear regularly in this Lucky Luke comics book. So if I'm going to present you each of these Daltons, and I will start with the less skillful Dalton, which is Avrel, and I compare it with Bacillus Weyenstephanensis, because not a big issue regarding to food safety, nevertheless, some abilities which might be interesting to know as a food industrial perspective. But Bacillus Weyenstephanensis is a psychotolerant bacteria. It has the ability to grow at low temperature, as low as 4 to 8 degrees Celsius. You have the ability to distinguish Bacillus Weyenstephanensis from the other members of Bacillus cereus group using PCR and targeted biomarker. Some strain may produce emetic toxin at temperature higher than 20 degrees. And Bacillus weyenstephanensis can be involved in food spoilage of dairy, ready-to-eat or egg-based products. The second one is William, which I associate in this presentation as Bacillus thuringiensis. This species has a large diversity of virulence factor. You have VIP protein, thuringiensin, parasporal body, and this is the one I want to focus in this presentation. This parasporal body, which is also named as crystal, this is a protein inclusion that is encoded by Cre and SID gene family. These crystals are produced during the sporulation, and these crystals show indicidal properties, and this is why it has been largely used for crop protection. The fact that these crystals are produced sporulation is quite important, because this is the only way at the moment to be able to distinguish between Bacillus thuringiensis and the other members of Bacillus cereus group. Different serovars have been reported and are used in the EC regulation. Kurstaki, Aizawi, Israelensis, and there is more than 300 commercial product formulation. So as I said, it is widely used as crop protection against caterpillar, beetles, fly, and mosquitoes. Bacillus cereus sensu stricto, so they are hemolytic and motile strain. They are involved in food poisoning due to temperature abuse and improper handling or storage condition. You have diarrheic strain, which are involved in food poisoning. Enterotoxin are heat labile. Different kind of enterotoxin are produced during growth in the small intestine, so once the food has been consumed. You have optimal production temperature of enterotoxin that are known. Similarly, regarding emetic strain, there are a lot of information already is available. They are often associated to the consumption of contaminated cooked rice, and this is due to cereulin, which is a heat-stable toxin, which is preformed in the food. Bacillus cytotoxicus is more distant cluster. They are heat-atorion strain with high virulence factor. It is possible to distinguish them from the other members of the Bacillus cereus group using specific markers, CK1 markers. Up to now, there are about 10 clinical isolates that have been characterized, but there is a high prevalence of Bacillus cytotoxicus in dehydrated ingredient, according to Consen publication. And finally, Bacillus anthracis, which is a highly monomorphic and clonal lineage of Bacillus cereus group. This is the etiological agent of anthrax, which is a zoonotic pathogen, maybe lethal for humans. And virulence factors are harbored on 2 megaplasmid. So as you see, Bacillus cereus strains show great genome plasticity, which is why these strains really adapt easily to all kinds of conditions. While certain virulent factors are located on large plasmid, genes that are encoding for diarrheic toxin are harbored by the chromosome, which makes it difficult to distinguish between pathogenic strain. At the moment, there is no possibility to distinguish diarrheic toxin strain. Regarding the methods, so different methods are available, commercial kits to detect a diarrheal toxin, which can allow for a semi-quantitative information as soon as the growth condition is well standardized. Pay attention that the kit are only targeting for specific part of the toxin and will not ensure the activity of the whole toxin. Regarding emetic toxins, there is no commercial kit, but there is an ISO method regarding the quantification of this toxin in food. Several genes that can be targeted to screen these pathogens. Regarding the method, there is one ISO method regarding the enumeration of presumptive Bacillus cereus colonies at 30 degrees. MYP is the reference medium agar. The colonies appear as pink because they don't have the ability to hydrolyze the mannitol. They have or do not have the precipitate, which is due to the lecithinase activity. So a food poisoning strain have this halo precipitate, but not the other. The confirmation step is performed in the second step on blood agar. You also have alternative method that are validated according to this ISO 16140 methods, which is from Baccarin and Compass agar, where you have specific colony that are appeared on the chromogenic media, so the colored colony are quite easy to read. No confirmation step is needed, so this is quite interesting as well. Another method which I found really interesting is the affiliation of phylogenetic group. This has been described by Guinebrotière in 2008 and 2010. There is one available tool that allows to compare, to blast sequences and to determine the group, phylogenetic group of Bacillus cereus isolates. This is based on partial sequence of PAN-C. PAN-C is found in a lot of microorganisms, but seems to be really discriminant in Bacillus cereus group. So all these more information are available on this link, where you see here different link related to the publication, more information on the phylogenetic group, sequences, protocols that are needed. So you just need to copy paste here the sequence, the PAN-C sequence of several isolates, then to run the blast, and you have the group that are associated to each isolated entered here. So quite easy, user-friendly tool. And this is giving some interesting information, because according to this work, 7 phylogenetic group may be determined. And for each of these 7 groups, you have different features, different phenotypic behavior. For instance, growth at low 10 degrees or 40 degrees, the heat resistance, and the implication in food poisoning. So you see here, you have group 3, which is highly involved in TIAC4 as well, and group 7. Regarding growth, you have here group 2 and 6 psychotroph tolerant strain, while the other are more mesophilic. You also have information regarding to the presence of genes. So as you see, chromosome, the gene encoding for enterotoxin are harbored by the chromosome, so you have the detection of all these genes for all the groups. One point which is interesting to note is that emetic strains are belonging to group 3. Not all strains belonging to group 3 are emetic, but emetic strains are only encountered in group 3. Here. And Cytotoxicus strain, highly virulent strain, also distinguished via the presence of this particular gene biomarker. When you associated the species to all of these 7 phylogenetic groups, you see that you got Bacillus cereus antiregensis in all these groups except group 1, Pseudomycoides, and group 7, Cytotoxicus. There are also methods to determine molecular fingerprint, which can be quite interesting to cluster some strain or to trace contaminant along the food chain. And now I would like to have a few examples regarding predictive microbiological based tool, in order to have some information regarding to food formulation. Is it supporting growth of Bacillus cereus? Is the process ensuring destruction of my contaminants, and is during shelf life the development of Bacillus cereus will occur. So in this case study, I just used rice pudding, where you have here the process to obtain this rice pudding, some information as well. So first step is this food formulation allowing growth of Bacillus cereus. Regarding the end product information that I got here, the pH and water activity, I am able to determine growth, no growth boundaries. Here you have the pH as a function of temperature for a specific water activity of 0.98. This is the result of the simulation of 9 strain, and you have here boundaries, which means that below this green line you have no growth of this Bacillus cereus strain. Above this line here, the combination of pH, temperature, or water activity will support growth. So in these two cases, if I got a produce which is at pH 6 but stored at 4 degrees or 8 degrees, you see that I am in the range allowing growth. Will a short change in the formulation impact these results? You see, if I change here the water activity, so the lines are a bit different. For case scenario 1, if my produce is stored at 4 degrees, I am in the safe area below this green line. If I'm not sure about the refrigeration condition, I am in the condition supporting growth. So unless strictly stored at 4 degrees, food formulation will support the growth of the microorganism. So the second step is then to check at the heat treatment. Is this heat treatment 90 degrees during 45 minutes will ensure the destruction of the strain? You have here the simulation of this population destruction. For strain 1, I will obtain 2 decimal reduction, but as soon as I will use another strain, which is a highly heat-resistant strain, you see that the same thermal treatment will reach much less efficiency, because here I got this 5 decimal reduction that I was targeting, and obviously the heat treatment will not ensure the destruction of this resistant strain. So you can play in a few clicks with such a tool, which are available, and this is quite interesting to click and to test different scenarios, because this is fast. This is easy to use. And if my last step, the third step, if I want to look at storage and distribution condition, because I'm not able to destroy all Bacillus cereus with heat treatment, how will my storage and distribution condition will impact on growth of the microorganism? So this, for this, I need one challenge test performed in my real food, my rice pudding. So this is a given combination of Bacillus cereus strain in rice pudding. I got then the growth rate determined in the food, and this growth rate can be used by decision-making tool to simulate the behavior for different conditions. So for instance, if my temperature scenario is well controlled, at the end of my storage time, which is 20 days, you will see that my distribution of my contamination will be much less than the targeted 5 log of contamination that I'm testing in this case. So no evolution of Bacillus cereus in the rice pudding, considering this storage scenario. But if I am using less stringent refrigerator condition, or if I have if I have a psychotolerant strain in the rice pudding, then the scenario might be completely different. So you have here the population distribution that is evolving after several days of storage. And you see that this population is evolving. And at the end of storage, the result is completely different. If I cannot ensure refrigeration condition, or if I have contamination with a psychotrophic strain, then my scenario is completely different. And in a few clicks, these decision-making tools enable to see and to predict the evolution of targeted population for different relevant scenarios. So finally, the take-home message. The first one is that Bacillus cereus group shows a wide range of behavior and adaptation ability. The second one is that food poisoning outbreaks are mainly due to temperature abuse and improper handling or storage of cooked food. And the third one is that there exist already available tools that may account for the diversity and may be used to simulate or to characterize a bit further the diversity and the contamination that can be found in the industry. So these tools, some are well known and are part of ISO standard, but the other one may be also under development. But I just wanted to show you in this webinar a few highlights, a few information that I wanted to share with you regarding this, this issue. And finally, I would like to acknowledge national funding, the project in BTID regarding Bacillus thuringiensis contamination. This is a project with Agri and Agro Food Industrial. And finally, all the members of this ISO working group, which are giving really interesting and sharing information regarding this, this issue. Finally, to acknowledge the team of this collaboration, Sporis collaboration. And I take the opportunity to announce you the symposium we're organizing in Quimper at the end of June regarding microbial food spoilage. And the program is now online, so please have a look at this website. And by this, I would like to thank you for your attention. And if you have questions, I would be glad to respond.
Thank you very much, Florence, for this nice presentation and the details you were showing to us. Maybe it's time now for the participants to ask a question. In the meantime, we have 21 people listening to your presentation. So maybe there might be some question they want to ask, so they just raise your hand and then I can open your microphone, or I can also make you a panelist, and then you can show your webcam. So are there any questions from the audience? Maybe I can ask a question in the meantime. These models you were showing, these models or these programs are available commercially, or these are tools which only you or your group are using?
Regarding the last example and the case study I presented, that these are part of available decision-making tool. This is not freely available. This is in previous tool, but there are other tools that are available, freely available, that can give answers, same kind of answer. These are based on mathematical modelings, which are recognized and accepted by the scientific public, and that are used for this kind of simulation. One particularity of SimPrevious is the generic approach I found really interesting, because it can be used for technological spoilage or pathogenic strain. And this is really applied to challenge tests, so perform in food. But plenty of other tools, predictive modeling tools, are available now on the market, freely or not, and these are really interesting to have a look, because this is giving a lot of information and this is gaining time.
Yeah. Thank you. We have a question now from Professor Marcel Zwintering from Wageningen University. So I will open your microphone. So your microphone is open now, so maybe you can ask your question.
Yeah, good afternoon, everyone. Can you hear me?
Yeah, perfect.
You can hear me? Okay. So first of all, thank you very much, Florence, for this very nice presentation. And I have a question that I'm sure you also don't have the answer to, but you will have some thoughts about the answer. And that's related to the sporulation conditions that you choose. If you look both, if you want to make predictive models of inactivation and of also the germination, but also if you want to investigate spore behavior, the conditions that you choose for sporulation are very important, but they have to be representative for reality. But of course, we don't know exactly where those spores are formed, so we don't know in reality what exact conditions we should choose in the lab. So if you would have exactly the answer how we should do it, I don't expect you to, because I don't think anyone has, but definitely you might have some ideas on that, on what things are to consider.
Exactly. As you said, the sporulation condition, for most of the time, they are not known. So in order to maximize risk, the spores that are produced in the lab are performed in optimal growth condition regarding temperature, pH. So this is yielding a lot of spore, and these spores show a really high resistance. If we would have performed the production of spore at lower temperature or lower pH, we would have got less spore population, and these spore would have been less resistant. So we are saying to our food industrial, okay, we're going to perform the spore in optimal condition for you to have a maximum risk. But as you said, depending of the application, depending on the topics to investigate, it might be interesting as well to have spore produced in less optimal condition. And these are part of the work that are run with our university partners, where we have spores, two kind of spore production in order to have an idea or to apprehend a bit the diversity of behavior we could have encountered. So we have spore produced in extreme condition, most of the time lower temperature, and spores produced in optimal temperature. But as you said, this is a really complex question. Thank you for asking, Marcel.
I can ask a second question unless there are other people that raise their hand. Gerhard?
No, at the moment you are the only one, so please go ahead.
So then this was on sporulation, and the next one is on growth. You did show those graphs where there were regions of growth and no growth, and in that graph there was a green line and a red line and another one, I don't remember the color, but with the green line you said if you are below this line then there is no growth. But if I looked at the legend, it said that the green line was 10% probability of growth, and so that is of course still for food industry a big issue, and even if it would be for product development. So with these type of growth, no growth models, how should, it's again a difficult question, what to choose, because it's of course it's not that black and white, but can you also give some considerations on that, on these growth, no growth ranges?
Yes, as you mentioned, these growth, no growth boundaries are really, to my mind, I'm not a predictive microbiologist, first of all. I'm dealing with these food predictive microbiology, so I'm now a bit familiar with the concept. What I found really interesting is that considering the combination of pH and water activity and temperature, you're able to identify conditions where you might have growth. As you mentioned, and perfectly right, these are probability of growth. So as soon as you are close to these boundaries, other impacts such as the texture, the presence of microflora, the presence of fatty emulsion, this kind of thing might really impact on the growth ability. So as soon as your food parameters are close to these boundaries, it is quite important to perform a challenge test in order to test in real life with real food if you will have growth or not. But if you are right below this green line or right above the red line, it's no use to perform challenge tests because you already have the answer.
Okay. Okay, thank you for the answers, and sorry for asking those questions in the gray area. And it really shows the interesting things of microbiology, going both from detection methods, genomics, characteristics, modeling, and of course also related to the quality and the safety of our food. So, bravo, well done.
Merci, Marcel.
So, any further questions from the others? So I think I don't have any written questions and there's no hand raised. Then I would like to say thank you again for your nice presentation. And I think I can also advertise that you will give another webinar on June 14, I think. Yeah. So there is another option where you can discuss with Florence on this microbiological topic. And if there are no further questions, then I would like to thank the audience and the people who contributed with questions, especially one. But still, if questions coming up, you can still forward the questions to me or directly to Florence, and you can maybe get an answer. So thank you very much again. And I will close now the webinar, and thank you for participating. And have a nice evening. Goodbye.
Thank you again. Thank you.